right? So with genetic crosses, you kind of have to like do crosses in order to get a really good hang of them. So that's why I wanna do some past questions with you guys before examination, right? But looking at genetic crosses now, we're just gonna describe them and see what the information, right, they give. How is that information useful? So here's our typical monohybrid cross that we've seen in CSET, where we're gonna have our different alleles, right, and we present with our genotypes, right, and then we mix based on this. So we know how to draw a Punnett square like this. So here's a Punnett square cross, so that's a bit better. Right, so the monohybrid crosses are fundamental tools used in genetics to study the inheritance of a single trait controlled by one gene with two alleles, right? These crosses are essential for understanding the principles of Mendelian genetics and predicting the outcomes of genetic inheritance in offspring, right? So this is what we're looking at. So that's really what we're doing by drawing a Punnett square or doing a genetic cross like this to figure out what the offsprings would look like, what percentage of the offsprings would be heterozygous versus homozygous versus homozygous dominant or homozygous recessive, right? Looking at what the phenotypic ratios would be versus the genotypic ratios. This is what we're doing, right? So you guys would have done this already in CSEC, right? So what do we use, right? So we can use a monohybrid cross, right, to predict the phenotypic and genotypic ratios of offsprings based on the genotypic and parental generation, the genotypes of the parental generation, right? So remember, by applying Mendel's law of segregation and dominance, we can determine the likelihood of different traits appearing in the offspring. Honestly, when we do our Punnett square, that what we're doing is applying the law of segregation and the law of dominance. That is what we're literally doing in our square. The law of segregation showing us different ways in which the alleles can be separated, which we do throughout the box and the quadrants. And then the law of dominance tells us, right, what is dominant over what and what the phenotypic trait will be. Right? We can also use it to test hypotheses, right? So monohybrid crosses are used to test hypotheses about the inheritance patterns of specific traits. So this is where the chi-square test would come in, right? So by comparing observed outcomes with expected outcomes based on Mendelian principles, scientists can evaluate the validity of genetic hypotheses and theories, right? It's also used in breeding programs, specifically so in agriculture and animal husbandry, monohybrid crosses are used to selectively breed individuals with desired traits. By crossing parents with known genotypes, breeders can actually produce offspring with specific phenotypic characteristics that are wanted, right? That are viable. Okay? And understanding genetic disorders as well. We can also use monohybrid cross to help us to understand genetic disorders, right? Well, genetic disorders are caused by single gene mutations. So by studying the transmission of the disorders through generations, researchers can actually identify the genetic mechanisms and develop strategies of diagnosis and treatment. So even though it seems so simple, monohybrid cross can do so much, right? For the dihybrid cross, however, right, we're using the general pea plant, Mendel's pea plant cross, right? So in the dihybrid cross, we know that involves the simultaneous study of two different traits controlled by, two different traits controlled by genes located on different chromosomes now. So far apart, far apart on the same chromosome, right? So what I was saying, is that the genes, right, the traits can be given by genes on different chromosomes, or genes are different are on different parts of the same chromosome. It could be on two different chromatids. It could be somewhere far away, right? But we're looking at how those genes exhibit different traits, right? These crosses provide insights into inheritance patterns of multiple traits and the principles of independent assortment. So this is what independent assortment is showing you. Showing you that when these genes are actually mixed up now, there are a lot more possibilities, right? So these dihybrid crosses are now exemplifications of meiosis, if you look at it like that, all right? So we can predict phenotypic and genotypic ratios from dihybrid crosses as well. We can map genetic linkages. So dihybrid crosses can be used to map genetic linkages between different genes and on the same chromosome. So by analyzing the frequencies of recombination events in offspring, Scientists can determine the relative distances between genes and create genetic maps of chromosomes, right? We can also study genetic interactions generally, right? When we look at that. So genetic interactions would be whether or not the genes are more likely dominant or epistatic, right? So dihybrid crosses provide insight into genes interactions such as epistasis and gene complementation. I know that we don't look at gene complementation in gate biology, right? We also have selective breeding, right? So we can also use dihybrid crosses for generally selective breeding that we discussed before for monohybrid crosses. So you guys need to know how to create or how to do dihybrid crosses. And in that case, we'll have to look at some past papers in order to go through this. Okay? Now into the chi-square test, our favorite part of module 2. Alright. Now, the chi-square test is a fairly simple statistical technique that we use, right? Chi-square tests are done all over science, by the way. It's not just biology, but we use a little bit of the biology, right? This is a part of statistical biology, right? Some amount of bioinformatics, right? So we're looking at a little bit of statistics, right? Biostatistics, right? So if you're gonna do like biology UE or something, like, you're gonna have to do courses called biostatistics, which we're gonna be looking at chi-squared tests, right? And chi-squared tests, trust me, is the least of our biostatistical problems. It's the easiest, right? So let's have a look at this now. We're looking at the chi-squared test. Look at independence, right, between alleles. So by definition, chi-squared, which is literally the Greek letter chi squared, right? The test, right, is a statistical test used to determine whether there is a significant difference between the observed frequencies of certain events and the frequencies that would be expected under a null hypothesis. So it is commonly used to assess the goodness of fit, right? The goodness of fit of observed data to the expected frequencies or the test or to test the independence of two categorical variables, right? So we're looking at the degree of independence, right, between two variables. Is one variable completely dependent on another? Is it epistatic? Is it not independent on each other? Is it non-epistatic, right? Does it agree with the null hypothesis, right? That there is no interaction, right? Or does it actually disagree with the null hypothesis, right? This is what we're looking at for the chi-square test. Any issues so far? I heard that this is a problem area, a little touchy topic. So if there are any problems, please stop me, right? So that we can clear up the mystery behind this, right? Now the null hypothesis. This hypothesis states that there is no significant difference between the observed and expected frequency. Any deviation between them is due to chance. Make sense? This hypothesis states that there is no significant difference between the observed and expected. The only differences in there are actually now due to chance. What is this saying now? What is it saying? Correlation does not equal to causation. That is what the null hypothesis is saying. Right? Correlation is not equal to causation. Meaning that because the two variables look like they're correlating somewhat or less, right? That does not mean specifically that one variable affects the expression of another. That does not mean that it is epistatic, but it could just be due to chance. That is what the null hypothesis is saying, right? Expected frequencies. These are the frequencies that would be expected if the null hypothesis were true, right? They are usually calculated based on theoretical probabilities or proportions. So what we do, we looked at what is there, and then we look at the genes and how they work, and then we draw a monohybrid or a dihybrid cross to figure out their expected frequencies due to the ratio of genotypic genotypes, right? The genotypic ratios, right? And that is how we draw our expected frequencies, and then we use it to plug it into the equation. All right, I'm looking at them in depth because I want to make sure that the topic itself is not an illusion. Right? For observed frequencies, there are the actual frequencies observed in the calculated data. So that's the frequencies that they give you. That's the observations that they give you. And from the observations that they give you now, and the expected frequencies from your monohybrid or dihybrid cross, then you're going to be able to calculate your chi-squared statistic. The statistic this statistic quantifies the difference between the observed and expected frequencies. It is calculated as the sum of squared differences between the observed and expected frequencies divided by the expected frequencies for all categories. What is this saying? We're gonna look at it, right? We are gonna have a look at it. The statistic for the chi-square, right? So it's calculated by the sum, the symbol of summation, right? So if you look at that, if we look at use the symbol of summation, let me draw the equation right here. So chi, right? So chi squared, the chi, is equal to, why that looks so, that's not supposed to be an E. This is sigma, by the way, guys. So sigma, right? Then we're gonna have E, I use E naught for this. E naught, so E original, right? Minus what is actually seen, right? What is expected, right? Divided by overall, right? The expected frequencies, right? And that should give you your value. Okay, that should be fine. All right, now if we look now at the degrees of freedom, now this represents the number of independent categories in the data. It is calculated as the number of categories minus 1. So if we have only 2 traits with minus 1 and degree of freedom is 1. If we have 5 traits and with minus 1 degree of freedom is 4. If we have 7 different traits, the degree of freedom is 6. If we have half a trait, degree of freedom is negative half. Right? So always minus 1. If you have 7 traits, the degree of freedom is 6. Right? So you minus 1 from the categories. Lovely. And that's how you get your degree of freedom. Significance level now. This is the threshold for determining whether the difference between the observed and expected frequencies is statistically significant. Commonly used significant levels include 0.05 and 0.01. So we'll look at whether or not it surpassed these levels to figure out whether or not we have an epistatic gene here. We have some amount of gene complementation or epistasis, right? Whether or not we break the null hypothesis. And if we're below now the threshold, we keep the null hypothesis. All right? So that's what's happening there. So here now we're gonna look at an example. Any questions or concerns? Before we go into the example, are there any questions or concerns? All right. Seems like there are none thus far. So our scenario here now, what our scenario is now is that, well, this is our scenario. So let's consider a scenario where biologists want to determine whether the observed frequencies of different flower colors in a garden match the expected frequencies based on Mendelian genetics. The biologist predicts, right, through a chi-square test or whatever, not the chi-square test, through a cross, right, a genetic cross, that we have a 3-to-1 ratio of purple, which is dominant, to white flowers based on the genetic makeup of the parent plants. So what we're going to do is actually just run through what we have here in this scenario. We do a test, right, as a Punnett square or whatever it would be, right, to figure out our ratio, right? So our ratio now would be our expected. So our variables now, we know what our null hypothesis is, right? What our null hypothesis will be is that there are no significant differences between the observed and expected frequency of flower colors in the garden. So there any deviation is then due to chance. Our expected frequency now is what we did through our square. So it's 3-to-1. The biologist expects that a 3-to-1 ratio is there. Our observed frequency now, right, is that after observing and counting the flowers in the garden, the biologist finds out that there are 100 flowers, 75 are purple, and 25 are white. So how do we do that now? Chi-squared is equal to this sigma, right, the complete sum, right, of the observed minus the expected squared divided by the expected. So here now, our chi-squared now would just be this. Our observed is 75, our expected is 75, divided by 75, and that squared divided by 75. Plus 25 minus 25 squared minus 25, right? What we're going to end up with is 0. So what does this 0 tell us from our chi-squared value? It tells us here, right? We're going to talk about that, right? But just for the degree of freedom, right? We know our degree of freedom is 1 because we only have purple or we have the other color, right? Or white. So since there are only two categories, purple or white, that the organism can exist in, our degree of freedom is 1. So two categories minus 1 is 1. Our significance level alpha now, what we're going to be using here is 0.05 as our significance level. And what we must note is that if we surpass the significance level, we must draw a new hypothesis, right? We must reject the null hypothesis. And if it is below 0.05, right, we must accept the null hypothesis. Remember, our chi-squared value now was 0. Therefore, it falls below the critical value or significance level of 0.05. We failed to reject the null hypothesis, and this means we ultimately accept the null hypothesis, saying that there is no significant difference between the observed and expected frequency of flower colors in the garden, and any deviation observed can be attributed to change. That is our conclusion, and that is what we're using our example to do. Right? That is the end of understanding our monohybrid and dihybrid crosses, the importance of those crosses, and using those crosses to interpret the statistical data in a chi-square test.